Hello students, how are you all? I hope so you are doing good. And let us enter into the class. Today we are going to discuss about the structure of genetic materials. Okay, what is genetic material? The genetic materials is either DNA or RNA. So it varies from the organism to organisms. Am I right? So examples, if a plant is having the DNA, in case of virus, it is having the RNA as the genetic materials. How do we know that DNA or RNA is the genetic materials? So we have discussed in the previous class about the Griffith experiment as well as the Asean and Chase method. What was the conclusion of Griffith experiments? So Griffith concluded that the heat-killed S strain bacteria transferred certain character to R strain bacteria. So something is transferred from the heat-killed S strain bacteria to R strain bacteria to make it virulent. So we know already that the S strain bacteria isn't virulent or causing pathogenicity, but R strain bacteria is a non-virulent. So in the previous class we have seen all these things in detail. Am I right? So the Griffith used E. coli bacteria as his experimental animals. He has used E. coli as his experimental organism. So that will be the key point for you. So what organism is used in the Griffith experiments? When the question is asked like that, you can answer it. E. coli is used in the Griffith experiments. Okay. So Griffith told the genetic material from the heat-killed S strain to R strain is transferred. Okay, that's why this R strain is transformed. The R strain is transformed into virulent form. So this is, we call it a transformation principles. But he was not aware of that, the biochemical assay. So he was not able to tell actually what is transferred, whether the protein is transferred or the genetic DNA is transferred or enzyme is transferred. So in order to clarify this doubt, so Avery et al. Groups has come. Okay, Avery et al. What he was doing? So he has conducted a series of experiments by taking out the extract from the heat-killed S strain bacteria, such as the extract a protein, that extract is an DNA, the extract is an enzymes. So all these things he has taken, and finally he has done the series of experiments. So he has concluded that the genetic material is DNA, because when he add DNAs to the extract and centrifuge, then what happens? Okay, so when he added the DNAs enzyme, okay, to the DNA, so this DNA degraded DNA. This DNA is degraded DNA. That means the DNA's enzyme degraded DNA so that the mice was alive. The mice was alive. So thereby he concluded the genetic material is DNA. Got it? Clear? So this can be proved evidently by Asean and Chase method. So what Asean and Chase used? They used a bacteriophage. They cultured the bacteriophage in two different isotopes, radioactive substances. One is phosphorus 32, another one is sulfur 35. Okay, so when he culture, after his culturing in the radioactive substances, the radio labeled bacteriophage are allowed to infect bacteria. The radio labeled bacteriophage are allowed to infect bacteria. When it infected the bacteria thought that this is its own genetic material, so started to multiply. So it multiplied the capsule as well as the genetic material. Okay, the capsule contains sulfur 35 and the genetic material contains phosphorus. So thereby he concluded DNA is having the phosphorus content, so thereby the genetic material is the thing which is just transferred from the organism to organism. So he has given the final conclusion and evident remarkable statement that the genetic material is DNA. Okay, so what is DNA? Let us see what is DNA. DNA is the polymer of nucleotide. Polymer of nucleotide. The full form of DNA is deoxyribonucleic acid. Deoxyribonucleic acid. Only the DNA will be the genetic material, not like that. So RNA also the genetic material. This genetic material present in viruses. Okay, so what happens to the DNA? How this DNA genetic material is composed of? We call the genetic material is a nucleic acid. The genetic material is a nucleic acid. So thereby this nucleic acid is composed of three main components. The first one is a pentose sugar, that is a ribose sugar. The second component is nitrogenous base, and the third component is phosphate. So all these three things, they are tightly bound to each other's link and forms a nucleotides. So the nucleotide is nothing but it is repeated units. The nucleotides, the DNA is nothing but the repeated units of nucleotide. That's why we call it a polymer of nucleotide. Okay, so what is the nitrogenous base? So what are nitrogenous base? Nitrogenous bases are two categories. One is a purine, another one is a pyrimidines. What are purines? Adenine guanines, they belongs to purine. Adenine guanine belongs to purine. But what about the remaining things? Pyrimidine. The pyrimidine will have the cytosine, thiamine and uracil. Cytosine and thiamine and uracil. The cytosine is common for both DNA and RNA. The cytosine is a common nitrogen base for both DNA as well as the RNA. But the uracil, okay, uracil is present in the RNA. Thiamine is present in the DNA. Okay, so the thiamine is replaced by the uracil. The chemical name of the thiamines is a 5-methyl-uracil. 5-methyl-thiamine. The 5-methyl-thiamine is the chemical name, other name of thiamines. So the uracil replacing the thiamine in RNA. Do you understand what I am saying? Clear? So what are the nitrogen bases? Two nitrogen bases. One is in purine, another one is in pyrimidine. So what is purine? Then adenine, guanine are purines. And what is the pyrimidines? You will say that cytosine, thiamine and uracil. Cytosine is a common for both DNA as well as RNA. But the uracil replace the thiamines in RNA. Do you understand? Okay, let's move on to the how this DNA polymers are formed. How these polymers of nucleotide are arranged. Let us see the structure of DNA. Before entering into the structure of DNA, we should remember three scientists. The scientist number one, Watson-Crick, as well as that Wilkins and Franklin Roosevelt. Okay, so Wilkins as well as Franklin Roosevelt. So these are the scientists. We should keep it in our mind. Why? Because Watson-Crick has made a model, DNA model, double helical structures of DNA. So who are all helping to this model? So what are the core concepts? How this DNA structure, the double helical structures are formed by Watson-Crick. They have followed certain basic principles from the Franklin Roosevelt and Wilkins. Okay, along with that Erwin Charkoff. Who are all these Franklin Roosevelt and Wilkins? So Wilkins and Franklin Roosevelt, they used X-ray diffraction in order to see the DNA structure. So with the help of the X-ray diffraction method and more core component is Erwin Charkoff rule. So with the help of Erwin Charkoff rule and X-ray diffraction method, that Watson-Crick, they elucidated the double helical structure of DNA. Okay, do you understand what I am saying? Clear? That's good. So when you see the nucleotide, I told you the DNA is nothing but polymer of nucleotide. What is nucleotide? What will be present in the nucleotide? Okay, So let us see what is present in the nucleotide. So look at this. This is the structure of the nucleotide. So here this is the ribose sugar molecule. Okay, and this is also ribose, double stranded structure. This is the double stranded DNA helix. Okay, so it starts from 5 prime to 3 prime, then this 3 prime to 5 prime, because they run anti-parallel to each other. If one strand start in the 5 prime to 3 prime and another strand will be 3 prime to 5 prime. Okay, the synthesis of the DNA always occurs in 5 prime to 3 prime. The synthesis of DNA always occurs in 5 prime to 3 prime. Okay, that I will be discussing in later classes. Let's move on to the structures. So here the nucleotide. So what is nucleotide? The nucleotide is having the components of ribose sugar, and to the ribose sugars what is attached? Along with the ribose sugar, that the nitrogen base is attached. Okay, nitrogen base is attached. Then with that the phosphate group. So the nucleotide will consist of ribose sugar, nitrogen base and phosphate group. All these things will be there. That's we call it a nucleotide. Okay, so here you see that this is the ribose sugar. To the ribose sugar, so we are having the carbon 1, carbon 2, carbon 3, carbon 4 and carbon 5. Okay, at 1C of ribose sugar, at the carbon 1 of the ribose, there is a nitrogen base. It is attached through the N-glycosidic bond. So the nitrogen base attached to the ribose sugar through the N-glycosidic bond. So this is we call it nucleoside. So what is nucleoside? The ribose sugar which attached with the nitrogen base, any one of the nitrogen base. So nitrogen base and ribose sugars form nucleoside. Okay, so the nucleoside name based on the what type of nitrogen is attached to the ribose sugar. If it is adenine, then adenosine or deoxyribonucleotides. Okay, if it is cytosine, then we call it cytidines or deoxycytosines. Okay, if it is thymine or thymidines, or we call it deoxythymosine. Okay, so based on the nucleoside is based on the nature of nitrogen base attached to the ribose sugar. I think I am not going faster, so keep up. So ribose sugar attached to the nitrogen base through the bond called N-glycosidic bond. N-glycosidic bond, where this adenine is attached to the ribose sugar. So for carbon C1 of ribose sugar, at the C1 of the ribose sugar the adenine is attached. Okay, so look at then where the phosphate group is attached. So the phosphate group is attached at the nucleoside 5th carbon. It's attached at the 5th carbon of the ribose sugar. Then this is where the phosphate group is attached. So this forms a nucleotide. This forms a nucleotide. So look at that. Okay, so this is we call it a nucleotide. Okay, so what is nucleotide? Now you are very clear. Nucleotide is composed of ribose sugar, nitrogen base and along with the phosphate group. Okay, what is nucleoside? The adenine or the nitrogen base attached to the ribose molecules, that's we call it nucleoside. Okay, the adenine attached to the ribose sugar at C1 carbon of ribose through the N-glycosidic bond. Then this phosphate group attached the C5 of the ribose sugar, so that forms then it's through the phosphodiester bond. Okay, this is one nucleotide. Okay, so mononucleotide. This mononucleotide attached bind with another nucleotide to form dinucleotide. Okay, the same way the ribose sugar 3rd carbon C3 of first nucleotide is attached to the 5th carbon of second nucleotide through the phosphate groups. So this is we call it phosphodiester bond. So one nucleotide is attached with bound with another nucleotide to form dinucleotide. It goes on finally forming polynucleotides or polymer of nucleotide. Do you understand? So adenine, I means the nitrogen base bound with the ribose sugar at C1 carbon of ribose sugar. Okay, so through the N-glycosidic bond. The second, then you are finding a phosphodiester bond. So where the phosphodiester bond is formed, that the fifth carbon of the ribose sugars where the phosphate group is attached. Okay, that forms a phosphodiester bond. So one nucleotide is attached with another nucleotide through the phosphodiester bond. It goes on. Do you understand? Clear? Got it. So thereby we can say that C1 of ribose, adenine is nitrogen base is attached. Okay, C5 of ribose, phosphate group is formed. Okay, got it? So this forms a nucleotide. So one nucleotide is attached with another nucleotide. That's an C3 to C5. So C3 of ribose sugar of one nucleotide, C5 of ribose sugars where the phosphate group is attached. So forming, so this forms and this is C3 to C5 attachment phosphodiester bond. Phosphodiester bond. Got it. So look at the structure of the DNA. This is a right-handed double helical structures of DNA. There are different types of DNA: bDNA, ddNA, Z and aDNA. So this is right-handed helical structures of DNA. So usually we call it an bDNA. We will be discussing later the types of DNA. Okay. So look at here. This DNA is having two strands. Okay, this strand are running to opposite, that is an anti-parallel, though they are having the 5 prime to 3 prime. How this 5 prime to 3 primes comes? Okay, here we are having the nucleotide type 1. You just consider this is 1. It is having the 5 prime in where the phosphate group is attached. Okay, then the nucleotide 2. Okay, so here then it is having the 3 prime end which is free where the hydroxyl group is attached. Okay, based on that only we are keeping it 5 prime to 3 prime. So free end of one nucleotide which is having the phosphate group and the free end of the nucleotide at the 3 primes having the hydroxyl group. So they are having that phosphate free end, hydroxyl free end. So this 5 prime to 3 prime. 5 prime to 3 primes. Do you understand what I am saying? So look at the structure again. Double helical structures. Okay. So see here, and this, how this sugar group, I means the sugar plus nitrogen base. Sugar group and nitrogenous base forms a backbone. Sugar and phosphate groups forms backbone of the strand. DNA strand. Okay. So this here we will identify the sugar and phosphate group. Like that it goes on. Sugar and phosphate group. So sugar and phosphate group forms a backbone. Okay. The nitrogen base project inward. Project inward of the strand. Got it? பாருங்க, இந்த DNA strandல நமக்கு சுகரும் பாஸ்பேட்டும் அந்த DNA strand உடைய பேக்போனா செயல்படுது. நைட்ரஜன் பேஸ் இருக்கு இல்லைங்களா, அது வந்து projected inside inward. Okay, இப்போ then we have to see what is the base pair rules, Charkov base pair rules. Okay, Elven Charkov base pair rule. So this adenine is there, no? It's a purine, am I right? So adenine always bind to thymine. Adenine always bind with thymine. That is pair with thymines. Okay, and this adenine and thymine, they are connected by the bond called hydrogen bond. So when we want to break the DNA strand, first we have to apply more temperature to break this hydrogen bond. De-annealing. First we have to break this hydrogen bond, then only we can separate out the strand. Afterwards we will go for another levels. Okay, so de-annealing, that is breaking the hydrogen bond, is easy when we apply more temperature. So when the temperatures are increasing, the bond in the DNA strand will be denatured. Okay, so adenine pair with the thymine always, okay, through the hydrogen bond, double hydrogen bond. Then we are having the cytosine always pair with guanine. Cytosine always pair with guanines. Then this is a triple bond and then hydrogen bond. Okay, so what Elven Charkov says, he says the purine is always equal to pyrimidine. Any testing organism, pyrimidine. So purine always equal to pyrimidine. Examples: purine. What is purine? Adenine. So pyrimidine, you can say T. Guanine, you can say C. Okay. So purine always approximately equal to the pyrimidines. Okay. So A is equal to T, G is equal to C. Got it? The same another concept he says. The ratio of adenine plus thymine is equal to guanine plus cytosine. The ratio of AT is always equal to the ratio of GC. So the ratio of AT is equal to ratio of GC. These are the two things is proposed by Elven Charkov rules. So first thing what I told, adenine is always paired with thymine, then cytosine always paired with guanines. Okay, the same way if you go for RNA, then it will be RNA instead of thymine, that uracil will be replacing. Do you understand what I am saying? Clear? So this is Elven Charkov rules. So Elven has proposed two basic concepts. One is the purine is approximately or trying to be equal always in the testing organism, and then the ratio of AT is always equal to GC, So this is the things of Elven Charkov rules. With the help of Elven Charkov rules, then Watson Crick has made a model. Okay, so here we are seeing that one turn of DNA helical structure contains 10 base pair. One turn of DNA helical structure contains 10 base pair. So one turn will have 10 base pair. Okay, so here we are having the two ditches, ridges. That is, we call it an major groove. The major groove. Then small groove, we call it a minor groove. Okay, so this one, the major groove, minor groove, and then one turn of the DNA helicals is approximately is having the length will be the 3.4 nanometer. Okay, so you will be another things you will be having between the two base pair. Between the two base pair, the distance will be 0 point. Between the two base pair, the distance will be two consecutive base pair. Between the two consecutive base pair, the distance will be 0.34 nanometer. 0.34 nanometers. So 1 nanometers is equal to 10 to the power 9 meters. 1 nanometer is equal to 10 to the power 9 meters. So 1 millimeters is equal to 10 to the power minus 3 meters. Got it? Clear? So the distance between two consecutive base pair is 0.34 nanometer. Okay? The whole turn of the 10 base pair, the distance will be 3.4 nanometer. 3.4 nanometers. So based on this distance between the two consecutive base pair and this one turn, we are able to find out that how many base pairs are present in the DNA as well as the length of the DNA. So we are able to easily identify the length of the DNA based on the number of base pairs present in it. So based on this, there are different calculating method is there. Let us solve this problem. Okay, I hope so you are understanding the concept very clearly. So let's move on. So here the length of the human DNA is 6.6 10 to the power 9 base pair. The length of the human DNA diploid condition. So haploid condition. What is the length of DNA? We can calculate the length of DNA, the number of base pair. Number of base pair multiply with distance between two consecutive base pair. Okay, this is the formula to find out the length of the DNA as well as to find out the number of base pair in DNA. Okay, got it? Clear? Fine. So here the length of the DNA, that is the human DNA in diploid condition, it is in 2n of human DNA is 6.6 10 to the power 9 base pair. 10 to the power 9 base pair. Got it? Okay, then it is in haploid condition. Surely it will be 3.3 into 10 to the power 9 base pair. 3.3 10 to the power 9 base pair. Got it? Clear? Clear or not? Okay, fine. So let's move on to the calculation part based on the length of DNA and Elven-Charkov rules. Elven-Charkov rules. Let's first see the length of DNA. There is a question given: if length of E. coli, if the length of E. coli is 1.36 millimeter, so calculate the number of base pair in a cell. So I hope so the question is very clear. If the length of E. coli is 1.36 millimeter, calculate the number of base pair in a cell. Calculate the number of base pair in a cell. So what is given? The length of the DNA is given. The length of DNA is equal to number of base pair between multiply with distance between two base pair, consecutive base pair. Okay. So length of the DNA is given 1.36 and we do not know number of base pairs, so consideration n and multiply with 0.34 10 to the power minus 9. 10 to the power minus 9. That is nanometer. No, 1 nanometer is equal to 10 to the power 9 meters. Okay, got it. So now how will you calculate? So n is equal to 1.36. Okay, so 1 mm is equal to 10 to the power minus 3. So thereby we will go for, it will come down 0.34 10 to the power minus 9. Okay, so the answer will be 4.6 10 to the power 6 base pair. When we do the calculations, we will get it 4.6 10 to the power 6 base pair. Do you understand what is that? Okay, if the length of E. coli is 1.36 mm, 1.36 mm, calculate the number of base pair in a cell. Calculate the number of base pair in a cell. Length of DNA. The length of DNA is equal to number of base pair and the distance between two base pairs. Okay, so absolutely we will get number of base pairs in the E. coli is 4.6 10 to the power 6 base pairs. Got it? Okay, fine. So another question is like this. If the length of, second question, length of DNA has 45,000 base pair, how many complete turn? How many complete turn will DNA molecule take? Okay, the length of the DNA has 45,000 base pair. How many complete turn will DNA molecules take? So what is, we know already, one turn contains 10 base pair. One turn contains 10 base pair. Am I right? Clear? The distance between two consecutive base pair is 0.34 10 to the power minus 9. Okay, so now that's not required. Now what we'll do it? So it has the length 45,000 base pair. 45,000 base pair divided by 10. So 0 cancels. So thereby we will be having 4,500 ton. 4,500 ton.